say.
Thanks, Lola. So I think we're at a really interesting and pivotal time in cancer therapy. In the 70s, we had chemotherapy and radiotherapy. In the 90s, we moved on to small molecule inhibitors. And now I think this is the decade of immunotherapy and cellular therapy, which I think is going to be transformational. And it's already transforming certain diseases, but as I hope you'll see later on, there's an enormous amount of capacity to extend these approaches to other diseases. And so I'm going to tell you a little bit about our work on a pediatric cancer leukemia called acute lymphoblastic leukemia. So I'm a hematologist, a bone marrow transplant doctor. ALL, acute lymphoblastic leukemia, you can see the leukemic cells up here, is the commonest cancer of childhood. There's about 400 kids a year in the UK diagnosed with this. And actually, it's one of the great success stories of modern medicine. In the 1960s, virtually no one was curable. Now we expect 85 to 90% of children to be cured. If you relapse after chemotherapy, then we can salvage patients with bone marrow transplant, where we give this huge dose of radiotherapy to ablate any leukemia that's still there, and then we rescue with somebody else's stem cells. But until about 10 years ago, if you relapsed after transplant, then you'd had the maximal dose of chemotherapy, radiotherapy it was possible to give, and then it was really time for palliative care. There was nothing more that could be done. And this picture shows a little boy that I looked after when I was a lecturer at Great Ormond Street. He was a baby when he first presented, and we treated him with chemotherapy for 2 years, and he did reasonably well. He went off, and then 3 years later, his disease came back. We treated him with more chemotherapy, gave him a radiotherapy-based transplant, and unfortunately, even after that, his disease came back and he died. And I saw an awful lot of kids like this, and it made me determined that these kids have been through an awful lot. Families have been through a huge amount, and I just really thought that we needed new therapies, and they had to be completely novel because we'd reached the ceiling of what we could achieve with chemo and radiotherapy. And so this is the approach that that we took. I started working on this with Karen actually during a postdoc in Baylor. And it's called CAR T-cell therapy. And the idea here is that we genetically modify the patient's own immune cells to recognize the leukemia. And we do this by putting in a new receptor into the T cells that enables the T cell to see the leukemia as different. It recognizes, in this case, a target called CD19. And when it sees a leukemic cell expressing CD19, it gets switched on and it will kill those cells. And more than that, it gets a signal to grow when it sees those leukemic cells. So these become serial killers that can kill tens of thousands of CAR T cells. And the beauty of this approach is that you can use it in any patient with ALL because all patients with ALL express CD19. And this is I'm sure you've heard of personalized medicine. This is the patient's own cells. It's a living drug that can persist and will target the leukemia without targeting the rest of the body. So without all the toxicities that we see with chemotherapy and radiotherapy. And in a proportion of patients, you'll get memory, and those T cells will persist and keep fighting the leukemia. So this is a schematic of the approach. So here's our T cell with its receptor, which is directed against, say, flu or another virus. And we use a modified version of the HIV virus to put in a new gene, which goes into the nucleus. And then the cell takes that DNA and expresses this new chimeric antigen receptor, or CAR, on the surface, which recognizes CD19. Now, when this new CAR T cell goes into the patient and it sees a leukemic cell that expresses CD19, it will kill it, and it will keep killing more and more of those leukemic cells. A number of groups, including our own, have been working on this approach for 15 years or so, this is the pivotal Eliana study, which was a multi-center study, which was the first study to show that you could use this approach in multiple centers across multiple different countries. And this is what's called a Kaplan-Meier curve, a survival curve. And you can see that almost all patients respond at the beginning, and then some of these patients relapse. At about a year or 2 years, the curve flattens off, and around 50% of patients are disease-free at 1 year. And so based on this, the FDA and the EMA approved this drug, which they call Kymriah, for relapsed refractory ALL in 2018. And at UCL, in collaboration with Martin Pule, we developed a different version of this CD19 CAR, which had low affinity for CD19, and we showed, it was very interesting, it seemed to allow the T cells to grow more in response to the tumour, and in mouse models it seemed to have better anti-leukemic activity. And so we then did a study with funding from Gosh CC and Children with Cancer at Great Ormond Street, where we treated 14 patients with terminal ALL who'd relapsed after chemotherapy and transplant. And of those 14 patients, 12 achieved complete remission. And again, our disease-free survival was around 46%. The interesting thing about this study was that it seemed much safer than the licensed drug Kymriah, and this was also seen in adult ALL by my colleagues at UCLH. And based on this, a biotech company called Autolus have licensed this CAR and tested it in what's called a pivotal study, and now that CAR T-cell product, which is called Obe-cel, has been approved for use in adult ALL, and we're currently doing the studies to get approval and licensing in pediatrics. So I wanted to, a picture paints 1,000 words, and I just wanted to show how effective this can be. So this is Austin, and he developed leukemia when he was 2, and had 2 years of chemotherapy, really complicated course, developed an infection which meant that he had to have colostomy, and despite that, he relapsed while he was on chemotherapy. So he was treated with more chemotherapy, and then had a radiotherapy-based transplant. This is a huge dose of irradiation. You wouldn't survive it unless you had a bone marrow transplant. And then at 14 months after the transplant, he relapsed again, both in the nervous system and in the marrow. So he had a second bone marrow transplant, and despite this, he had a further relapse in his nervous system. So this child had 6 years of chemotherapy and 2 bone marrow transplants with lethal doses of chemotherapy and radiotherapy and all the side effects that entailed. And then we gave him, so he was the first patient enrolled on the CARPAL study, and this is him receiving his CAR T cells back in 2018. And here he is now, well, a couple of years ago. He's now 16, so he's almost 9 years after his CAR T cells. And he's in complete remission. He's very well. The only thing is he's quite short, because of the radiotherapy he had when he had his transplant. So what I'm trying to show is that a single syringe of 50 million cells was able to do what a lethal dose of radiotherapy and 6 years of chemotherapy was not able to do, and virtually no side effects. He had a fever at day 14 for about 4 hours, and that was it. while the data with Kymriah and our own CAR are extraordinary, it's important to realise that these are just the first iterations of this therapy. And around 50% of patients who respond will ultimately relapse. And there are two main causes of relapse. The first is the CAR T cells need to persist for at least a year. If they don't, then the patient relapses. And the second is sometimes the leukemia evolves to evade the CAR T cells. And so much of my research over the last few years has focused on how we can overcome these two key challenges. So let's start with overcoming antigen-negative relapse. So one thing we learned quite early was that patients who downregulate CD19 on their leukemia so it can't be seen by the CAR T cells still express other markers like CD22. And so we thought, well, maybe a single leukemic stem cell is unlikely to downregulate both CD19 and 22 simultaneously. So maybe, just like with multi-drug resistant TB or HIV, maybe targeting two antigens rather than one might be more effective. And so we did an initial study of this, cohort 3 of the CARPAL study, and we developed an alternative approach where we used two different viruses to infect our T cells, one expressing CD19 CAR, one expressing CD22 CAR. And so what you end up with is some cells expressing CD19 CAR, some cells expressing CD22, and the majority expressing both. And then we took this to another clinical study, and we treated 12 patients. It was very safe. 10 out of 12 patients went into molecular complete remission. The disease-free survival of the year was 60%. The most important thing was that there was no antigen-negative relapse. So all the relapses were positive for both CD19 and CD22. And we think this reflects the fact that we're using this best-in-class CD22 CAR. But we still haven't improved that survival by much, because the patients are still relapsing because of loss of CAR T cell persistence. And that's what I've been focusing on more recently. So what causes the early loss of CAR T cells? And it's extraordinary given the amount of money that's been put into this field and effort that's been put in this field. We don't really understand that. We think that the fitness of CAR T cells is important. Sometimes CAR T cells, when they see too much leukemia, get overactivated and exhausted. And sometimes there may be a component where the patient's immune system rejects the CAR T cells. And so we're studying this prospectively in a study called CARPAS. When we do these clinical studies, we try to learn as much as we can from them. And one of the things that we found from our previous study was that CAR T cells expressing both CARs, both the CD19 and CD22 CARs, may be prone to overactivation and to die because of that. And so we thought, well, maybe it would be good to separate the infection so that you make CD19 CARs and CD22 CARs, but nothing that's expressing both, and then pool them when you infuse. And then we also found that there's that the long-term persisting CAR T cells, 3-4 years out in patients, were derived from an early T cell compartment called memory T cells, and that if you shorten the way you make the cells, instead of going for 9 days, you make them over just a 5-day process, then you preserve these early CAR T cells. So this is our old process where we only had 3% of these stem cell memory T cells. Now we get 30% of these stem cell memory T cells. And we're combining this with 2 other measures to try and improve CAR T cell persistence. I mentioned that if you have persistent stimulation by the leukemic cells, the CAR T cells become exhausted and ineffective. So in the next study, we're giving a second dose of CAR T cells after the first dose has done the heavy lifting. And so we give that second batch of T cells, and they're not going to see as much leukemia, and therefore less likely to become exhausted. And then some colleagues in the US and Utrecht have shown that if you optimize the chemotherapy, which is called fludarabine, that we give before CAR T cells, that improves their persistence and decreases relapse. And so we're going to measure the levels of this drug, fludarabine, to make sure we get the right dose to allow the CAR T cells to expand. And we're also going to add a low dose of radiotherapy, very small dose, but that may help prevent CAR T cell rejection. And so in our next study, which is funded by CRIS Cancer, cohort 4 of CARPAL, we're hoping to overcome those two key challenges I started with in a comprehensive approach, incorporating dual antigen targeting with our best-in-class CD22, and trying to improve CAR T persistence by shortening manufacture to increase the stem cell memory T cells, separate transduction to prevent activation-induced CAR T cell death, optimized lymphodepletion with measuring fludarabine doses and a low dose of irradiation, and a split dose to try and prevent exhaustion. Now, as Marta said at the beginning, pediatric cancer is rare. You know, we're quite successful at treating pediatric ALL, and so the number of children that we can treat with this approach is 20, 30 in a year at Great Ormond Street. And so commercial companies are interested in that because there's already a product on the market, and why would they invest millions in developing a new product that might be better? And so it has been challenging to get funding for this indication, and I'm incredibly grateful to CRIS Cancer and to GOSH Charity, who funded this new cohort of the study. And I'm pleased to say we started the study in March. The first patient is now 2 months out and in molecular complete remission. Next patient's due to be treated in a couple of weeks, and the third patient we're about to make the cells on. And none of that would be possible without the funding from CRIS Cancer and GOSH CC. So just to finish, I think these engineered T cells, targeted specificity, are going to revolutionize cancer therapy. This particular study addresses the two key challenges for CAR T cell therapy in ALL, and we hope it'll allow more patients to be cured. But just as important, the lessons that we learn from these bench-to-bedside studies in leukemia are going to facilitate extension to solid cancers, which Antonio and Karen are going to talk to you about. And just to say once again, that I'm hugely grateful to CRIS Cancer for enabling us to deliver these groundbreaking new therapies to children who otherwise have no hope. So thank you very much for your attention, and thank you very much for supporting CRIS Cancer.